Okay, let's unpack this. Have you ever really thought about how a creature like, say, the star-nosed mole, with that incredible star-shaped snout, manages to coordinate all those tiny, rapid movements it makes underground?
Hmm,
How does it actually function as a mole, as a whole organism, reacting and anticipating, and not just, well, a pile of cells?
that's a great way to put it.
Today, we're diving deep into exactly that, exploring the amazing systems that let animals control and integrate their functions, adapt, and even keep track of time.
Yeah, it's a truly fascinating area. And that star-nosed mole, it's actually a perfect example of the kinds of physiological mechanisms we're going to get into.
Ah, okay.
We're drawing our insights today mainly from Animal Physiology by Hill, Wise, and Anderson. Really solid text. Our mission, if you like, for this deep dive is to understand these core processes, compare the different strategies animals use.
Which can be pretty clever, right?
Oh, incredibly clever. And also appreciate why they matter in the real world, their adaptive significance, and maybe even touch on how scientists figure this stuff out.
Excellent. So let's start with the big one, the system that really orchestrates so much of animal behavior, the nervous system. Um, at its heart, what exactly is a nervous system? What makes it so central?
You can think of it as the animal's master communication network. Really, it's a highly organized collection of specialized cells, neurons, and their essential support cells.
Right.
And they're all designed to transmit rapid electrical signals. These signals, they act like a biological internet, essentially,
Mm-hmm.
carrying information from sensory cells through processing centers,
Mm-hmm.
and then finally out to what we call effectors, like muscles or glands,
Mm-hmm.
which then carry out the actions. It's the ultimate system for, quick responses and complex coordination.
Okay, so it's this network of specialized cells. How are they organized? What are the main pieces of this system?
Well, broadly speaking, we divide it into two main parts. There's the central nervous system, the CNS,
Okay.
and the peripheral nervous system, the PNS. The CNS is the command center, the brain and spinal cord. That's where the main processing and integration happens. It's packed with neurons doing that work.
And the PNS.
The PNS is basically everything else, all the nerves stretching out from the brain and spinal cord,
Yeah.
connecting the CNS to the rest of the body, the limbs, organs, skin.
Got it. Linking it all up.
Exactly.
Yeah.
And within these systems, you have different types of neurons. Sensory neurons bring information into the CNS from your eyes, ears, skin, etc.
Right.
Motor neurons carry commands out from the CNS, telling muscles to contract or glands to secrete.
Okay.
And then, really importantly, you have interneurons. These guys are entirely within the CNS, and they act as the connectors, the integrators, the pathways between other neurons. Crucial for processing.
So they do the thinking, kind of.
In a way, yes.
Yeah.
They form the complex circuits. And just for terminology, when we talk about bundles of the neuron wires, the axons, the PNS, we call them nerves. Inside the CNS, similar bundles are called tracts, or sometimes commissures or connectives.
Okay, that clarifies things. Can you walk us through a concrete example? Like, how did these parts actually work together for a simple action?
Absolutely. Let's take the cockroach startle response.
Mm-hmm.
It's a classic.
Fast.
Very fast. So imagine a puff of wind hits the little sensory hairs, the cerci, on its abdomen. Sensory neurons there immediately detect that.
Okay.
They send signals racing into the CNS. There, those signals excite specific giant interneurons. These are specialized for speed.
Ah.
These interneurons then rapidly activate motor neurons.
Mm-hmm.
And those motor neurons send commands straight to the cockroach's leg muscles. Exactly. An almost instantaneous reflexive dart away from potential danger. It's a beautiful illustration of that core flow. Sensory input, CNS integration, motor command output, and then the effect of the muscle producing a coordinated adaptive response.
That's incredible. Such speed and precision from a simple circuit. And, what's really interesting is that these nervous systems, they aren't all built the same across the animal kingdom, are they? They vary hugely.
Massively.
So what does evolution tell us about how these complex systems actually developed?
Yeah, that's a huge question. Evolution shows some clear trends, especially in animals with bilateral symmetry, a left and a right side.
Like us.
Like us, exactly.
Mm-hmm.
We see two major patterns. First, centralization. This is the tendency for neurons, especially those doing the processing, to gather together into central clusters or cords, rather than being scattered like a simple net.
Makes sense. More efficient.
Probably, yeah. And the second trend is cephalization. That's the concentration of nervous structures, the brain, major sense organs at one end of the body, usually the head end.
The end that meets the world first.
Precisely. It allows for better processing of information about the environment the animal is moving into.
So looking across different groups, what are the main ways these centralized systems are organized?
Well, if we look at the big picture, we see sort of two main blueprints for central nervous systems. In arthropods, think insects, spiders, crustaceans.
Mm.
You typically find what's called a ganglionic nervous system. It features a nerve cord that usually runs along the animal's belly, the ventral side, and it's typically solid.
Yeah.
This cord is often structured like a chain of segmental ganglia, little clusters of neurons linked together, almost like a ladder. Each ganglion kind of manages its own body segment, with neuron cell bodies on the outside and a dense core of connections, the neuropil, on the inside.
A bit like distributed processing.
You could say that, yes. Now, vertebrates, including us, have a very different setup. Our central nervous system is a dorsal, hollow nerve cord, the brain and spinal cord. It runs along our back and develops from a tube structure in the embryo.
Dorsal and hollow versus ventral and solid. Big difference.
Huge anatomical difference. But here's what's amazing. Despite these differences, recent work, especially looking at gene expression.
The genetic toolkit.
Exactly. It strongly suggests a common evolutionary origin. There's even evidence hinting that our body plan, including the nervous system position, might be sort of flipped compared to many invertebrates.
Yeah.
Built from similar genetic instructions, just organized differently.
Wow. So the basic building blocks, the neurons themselves, are quite similar, but the architecture changed.
That's the key takeaway. The neurons function in fundamentally similar ways across vastly different animals. It's the organization, the way they're wired together into circuits and larger structures, that has evolved and become incredibly complex in diverse ways.
That is a profound idea, a shared heritage deep down. Okay,
Yeah.
let's zoom in on the vertebrate system then, particularly the brain. It's just this marvel of organization.
Yeah.
What are its main structural features?
Right. Inside the vertebrate CNS, the brain and spinal cord, we find two main types of tissue visually. There's gray matter,
Okay.
which is densely packed with the neuron cell bodies and all the synapses, the connection points. This is where the main computational work, the processing happens.
Thinking stuff.
Pretty much, yeah. Then there's white matter. This is composed almost entirely of myelinated axons, the long wires connecting neurons, wrapped in fatty insulation.
Myelin sheath makes signals go faster.
Exactly. White matter acts like the high-speed information highways,
Hmm?
connecting different regions of gray matter. And functionally, we usually divide the brain into three major regions: the forebrain, midbrain, and hindbrain, each with specialized roles that have become incredibly elaborated over evolutionary time.
And within this complex structure, how does it actually organize itself to do all the things it does? Are there fundamental principles at play?
Yes, absolutely. Neuroscientists have identified about 5 core principles that really help us understand vertebrate brain organization. They offer some powerful insights.
Okay, let's hear them.
First up, brain function is largely localized. Now, this doesn't mean the brain isn't interconnected. It definitely works as a whole. But specific areas are highly specialized for particular jobs.
Like different departments in a company.
Sort of, yeah. Think about language. Broca's area is crucial for forming grammatical speech, while Wernicke's area is vital for understanding spoken language. They're distinct areas, though connected. And modern tools like fMRI,
Brain scanning.
right, they clearly show different parts lighting up with activity during specific tasks. It beautifully demonstrates this functional specialization, although it's usually circuits, not just single spots.
Okay, localization. What's next?
Principle 2. Brains have maps. Our brains create internal representations, literally maps, of the outside world and our own bodies.
Maps. How so?
Well, take your somatosensory cortex, the part that processes touch. Different parts of your body project signals to specific regions there. This creates what's called a somatotopic map, sometimes visualized as a homunculus, a little distorted person mapped onto the brain.
The one with the huge hands and lips.
That's the one. Those areas are disproportionately large on the map because they have more sensory receptors and are incredibly important for how we interact with the world. It reflects functional importance, not physical size.
Fascinating. Principle 3.
Size generally matters. Now,
Right.
this is a bit nuanced, but generally, if you connect this to the bigger picture, a larger brain area or larger brain overall relative to body size usually means more neurons dedicated to that function.
More processing power.
Essentially, yes.
Yeah.
More neurons allow for more complex integration and processing. It helps explain why, say, a mammalian brain with billions of neurons can handle things like abstract thought and language, while an insect brain with perhaps thousands excels at different, maybe faster, tasks suited to its niche. It's about matching neural capacity to ecological needs.
Okay, makes sense. Number 4.
Number 4 is a key evolutionary insight. Vertebrate brain evolution has largely involved repeated expansion and reorganization of four brain areas. Evolution didn't necessarily invent brand new structures out of thin air all the time.
Right, it tinkers.
It tinkers, exactly. Many different vertebrate groups independently expanded and refined existing parts, especially in the forebrain. The development of the complex neocortex in mammals is a prime example. This allowed for new, more sophisticated behaviors and adaptations.
So building on existing foundations.
Precisely. And the fifth principle, which is truly remarkable, neural circuits are plastic.
Plastic, meaning they can change.
Exactly. Our brains aren't rigidly hardwired like a computer circuit board. The connections between neurons, the synapses, are incredibly dynamic. They change strengths, they form, they disappear throughout life based on development, maturation, and crucially, experience and learning.
Ah, so that's how we learn and remember.
That's a huge part of it. Long-term memories are thought to be stored as changes in synaptic strength. And there's even evidence now for neurogenesis, the creation of new neurons in certain parts of the adult brain. That's why stem cell research holds such promise for conditions like Parkinson's or Alzheimer's.
Wow. That's incredibly hopeful. So these 5 principles, localization, maps, size, forebrain expansion, and plasticity, they really paint a picture of a dynamic, organized, and adaptable brain. And you mentioned our star-nosed mole earlier. How does its unique brain fit into this?
Oh, the mole is a fantastic case study. It exemplifies these principles perfectly. Localization and maps. Its star, that amazing touch organ, has not one, but 3 distinct representations in its somatosensory cortex.
3 maps for the star.
Yes, and together, these maps take up more cortical real estate than the map for the rest of its entire body combined.
Whoa!
That's extreme cortical magnification. It directly reflects how vital that star is for feeling its way around and finding food in the dark underground. It's compensating for its poor eyesight. And size matters. That huge area means immense processing power dedicated to touch from the star. And yes, studies show its brain exhibits plasticity too, adapting its maps based on experience.
It really drives home how brain structure is shaped by lifestyle and sensory needs. Okay, fascinating stuff on the CNS. Let's move outwards now to the peripheral nervous system, the PNS. You said it connects the CNS to everything else. How is it divided up?
Right, the PNS acts as that crucial bridge. We generally divide it into 2 main functional systems, the somatic nervous system and the autonomic nervous system.
Okay, somatic first.
The somatic nervous system is largely what you think of as under conscious or voluntary control. It controls your skeletal muscles, the ones you use to walk, talk, wave your arms, pick things up.
Okay, voluntary movement.
Exactly. It also handles incoming sensory information from your external environment that you're usually aware of: touch, pain, temperature, vision, hearing, taste, smell. A key feature of its motor output is that typically a single motor neuron runs directly from the CNS all the way to the target skeletal muscle fiber.
A direct line. And the other side, the autonomic.
The autonomic nervous system, or ANS, is kind of the automatic or involuntary control system. It manages all your internal bodily functions that you don't consciously think about most of the time.
Like heart rate, digestion.
Precisely. Heart rate, digestion, breathing rate, blood pressure, pupil dilation, sweating, the action of smooth muscles in your organs and blood vessels, and the function of glands. It keeps the internal environment stable.
The unsung hero.
Definitely. And a key anatomical difference from the somatic system is its typical wiring. It usually uses the 2-neuron relay to reach its target.
2 neurons, how does that work?
There's a preganglionic neuron whose cell body is in the CNS. Its axon extends out and synapses with a second postganglionic neuron in a cluster called an autonomic ganglion, located somewhere out in the periphery.
Yeah.
It's the axon of this second neuron that then travels to the target organ, like the heart or the gut.
Like a little relay station. Why the 2 steps?
It allows for more complex control and integration. One preganglionic neuron can influence multiple postganglionic neurons, diverging the signal, or multiple preganglionic neurons can converge onto fewer postganglionic neurons. It adds flexibility.
Okay. And within this autonomic system, are there further divisions?
Yes, there are 3 main divisions we talk about. First is the sympathetic division. This is famously known as the fight-or-flight system.
Ah, adrenaline rush time.
Exactly. It gets the body ready for action, for emergencies, for exertion. It generally increases heart rate, raises blood pressure, diverts blood flow towards muscles and away from digestion, dilates pupils, mobilizes energy stores. Its preganglionic neurons originate in the thoracic and lumbar regions of the spinal cord, the middle part,
Okay.
and they typically synapse and ganglia located relatively close to the spinal cord. The postganglionic neurons then travel longer distances to the organs, and they primarily release the neurotransmitter norepinephrine related to adrenaline.
Got it. Fight or flight. What's the counterpart?
That would be the parasympathetic division. This is often called the rest and digest system.
Calming things down.
Precisely. It promotes functions associated with a calm, relaxed state, slowing heart rate, lowering blood pressure, stimulating digestion and absorption of nutrients, constricting pupils, conserving energy. Its preganglionic neurons originate from the brainstem, cranial nerves, and the sacral region of the spinal cord, the top and bottom ends.
Okay.
And their preganglionic neurons are typically long, traveling almost all the way to the target organs before synapsing in ganglia, located very close to, or even within, the organ walls. These postganglionic neurons primarily release acetylcholine as their neurotransmitter.
So sympathetic and parasympathetic often have opposite effects.
Very often, yes. They work in tandem, providing a balance, like an accelerator and a brake pedal for many bodily functions, allowing for really fine-tuned control depending on the situation.
Accelerator and brake. Good analogy. You mentioned 3 divisions. What's the third?
The third one is fascinating. The enteric division. This is sometimes called the second brain.
The second brain.
In the gut.
In the gut, yes. It's this incredibly extensive and complex network of neurons located entirely within the walls of the digestive tract from the esophagus down to the anus.
Wow.
It contains a huge number of neurons, some say more than the entire spinal cord. And it largely controls the moment-to-moment activity of the gut, like coordinating muscle contractions for peristalsis, controlling secretion, managing blood flow, all relatively autonomously.
So it runs the digestive show mostly on its own.
To a large extent, yes, although the sympathetic and parasympathetic systems can still influence it, modulating its activity up or down. But the enteric system has its own intrinsic reflexes and processing capabilities. It's a really sophisticated piece of neural machinery dedicated to digestion.
That's amazing, a whole nervous system just for the gut. Okay, so we've covered how the nervous system allows animals to react. But you mentioned earlier they also anticipate. That brings us to biological clocks, right? Exactly.
Yeah.
This is another layer of control, moving beyond simple reaction. Animals don't just respond to what's happening now. They have internal physiological timing mechanisms, biological clocks, that allow them to predict and prepare for recurring environmental changes.
Internal clocks, like the one that tells us when to sleep.
That's the most familiar example,
Yeah.
yes. These clocks create rhythms in almost everything. Sleep-wake cycles, body temperature, hormone levels, metabolism, activity patterns, you name it. And the key thing is they are endogenous.
Endogenous, meaning they come from within, not just reacting to sunrise and sunset.
Precisely. They generate the rhythm internally. There's this groundbreaking experiment decades ago. A man lived in a bunker, completely isolated from all external time cues, no clocks, no daylight, nothing.
Okay, what happened?
His physiological rhythms, body temperature, urination patterns, sleep-wake times, they continued. They kept cycling in a regular pattern.
Even with no outside information.
Even with none. But here's the crucial bit. His cycle wasn't exactly 24 hours. It free ran, typically becoming a bit longer each day, maybe 25 hours or so. This proved the clock was internal, endogenous, but needed environmental cues to stay synchronized to the actual 24-hour day.
Wow, so that internal cycle, that's the circadian rhythm.
Yes, circadian means about a day. That free-running rhythm generated internally is the circadian rhythm. But not all daily rhythms we see are truly circadian. Some might just be direct responses to light or temperature changes.
Okay, so if the internal clock isn't exactly 24 hours, how does it stay, on time with the real world? You mentioned environmental cues.
Right. The internal clock gets entrained or synchronized by external cues.
Yeah.
The German term often used is Zeitgeber, which means time giver.
Zeitgeber, okay.
And the most powerful Zeitgeber for most organisms by far is the daily cycle of light and dark. Think about a bird like a chaffinch. In a normal light-dark cycle, its activity starts right at dawn and ends at dusk, perfectly synced. But if you put that same bird in constant dim light, its activity rhythm continues, but it starts to drift, maybe beginning activity every 23 hours instead of 24. It's free-running. The light cycle normally resets it each day.
So the light acts like a daily adjustment for the internal clock. Where is this master clock physically located, at least in mammals like us?
In mammals, the master circadian pacemaker resides in a tiny pair of structures deep in the brain, in the hypothalamus, called the suprachiasmatic nuclei. or SCN for short.
Yes, Ian.
Yeah, they sit right above the optic chiasm where the optic nerves cross, conveniently positioned to receive light information directly from the eyes.
Ah, direct input from the light sensor. Clever. How do we know the SCN is the master clock?
There's some really elegant experimental evidence. For example, classic studies in hamsters. If you surgically destroy the SCN in a hamster, it completely loses its circadian rhythms. Its activity becomes random throughout the day and night.
Okay, that's strong evidence.
But here's the clincher. There are mutant strains of hamsters that have naturally shorter or longer free-running rhythms, say 20 hours instead of the usual 24-ish. If you take the SCN from one of these 20-hour mutant hamsters and transplant it into the brain of a normal hamster whose own SCN was destroyed,
What happens?
the recipient hamster adopts the 20-hour rhythm of the donor SCN.
Wow, that's incredible proof. The SCN carries the timekeeping information.
Absolutely. It really highlights the SCN's role as the central pacemaker coordinating rhythms throughout the body.
And how does it actually keep time? Like at the molecular level, inside the SCN cells, what's the mechanism?
It's incredibly elegant, actually. It's based on a molecular feedback loop involving gene expression. Think of it like a tiny genetic oscillator or pendulum.
Okay.
Basically, certain clock genes get turned on and produce specific proteins. These proteins build up in the cell cytoplasm. After some time delay, these proteins move back into the nucleus and act to turn off the very genes that produce them.
Ah, negative feedback loop.
Exactly. Then, as those proteins gradually degrade, the inhibition is lifted, and the clock genes turn back on, starting the cycle all over again. This whole cycle of gene activation, protein production, inhibition, and protein degradation takes roughly 24 hours to complete.
Like a tiny self-regulating molecular clock in each cell.
That's pretty much it. A rhythmic rise and fall of specific gene products, creating a stable, self-sustaining oscillation that keeps time.
That's really neat. So, okay, animals have these internal clocks synchronized by light. Why? What's the big advantage? Why is anticipating things so important for survival?
The major adaptive advantage is that predictive power we talked about. It allows animals to prepare for predictable environmental changes before they happen, rather than just reacting after the fact.
Like getting ready for dawn before the sun actually comes up.
Exactly. Think about our star-nosed mole again. Even deep underground, with no direct light cues, its internal clock can help it anticipate daily cycles, maybe when its prey is most active near the surface, or when it's safest to rest. It allows physiology and behavior to be optimized for the time of day.
Okay, anticipation. Are there other advantages?
Oh yes. Biological clocks allow animals to time crucial processes, even when the relevant environmental cues might be subtle or unreliable on a given day. They're also essential for measuring the length of the day, the photoperiod.
Photoperiod, how's that help?
Measuring changes in day length is how many animals time seasonal events. Things like reproduction, migration, hibernation, changes in coat color. They're often triggered when the photoperiod crosses a certain threshold, telling the animal what time of year it is. The clock is used to measure the duration of light or dark.
Ah, linking daily time to yearly cycles. Clever. Any other uses?
Yes. Some animals use their internal clock for navigation. Homing pigeons, for example, can use the position of the sun as a compass, but to do that accurately, they need to know the time of day.
Right, because the sun moves across the sky.
Exactly. If you experimentally clock shift, a pigeon basically reset its internal clock by manipulating light cycles in the lab and then release it, it will fly off in the wrong direction, misinterpreting the sun's position based on its faulty internal time.
Wow. So the clock is critical for their GPS. That's amazing.
It really is. And one more really cool property of these clocks, they are remarkably temperature insensitive, or more accurately, temperature compensated.
Meaning?
Most biochemical reactions speed up significantly as temperature rises. If biological clocks were like typical metabolic processes, they'd run much faster when an animal's warm and slower when it's cold, making them useless as reliable timers.
Right. A clock needs to keep steady time.
Exactly. So through various molecular mechanisms, biological clocks maintain a relatively stable period, a consistent rhythm, across a wide range of physiological temperatures. This makes them reliable timekeepers, even for animals whose body temperature fluctuates.
That's a critical feature. Okay, so we focused on circadian, about a day, rhythms. Do animals have other kinds of internal timekeeping, longer or shorter cycles?
Absolutely. Besides circadian clocks, there's good evidence for circannual clocks, generating rhythms with a period of about a year. These likely underlie some seasonal behaviors like migration readiness or hibernation timing, even in animals kept in constant laboratory conditions for years.
Yearly clocks. Okay.
And for animals living in coastal areas, there are circadial clocks, synchronized with the roughly 12.4-hour cycle of the tides. Think of fiddler crabs becoming active just before low tide to forage on the exposed mudflats. Their activity rhythm persists even if you take them into the lab away from the ocean.
Matching the tides makes sense for them. Anything else?
Yes, there are also what are sometimes called interval timers or hourglass timers. These aren't continuously cycling clocks like circadian rhythms, but mechanisms that measure a specific duration of time.
Like an egg timer.
Kind of, yeah. For instance, after a pair of pigeons lays eggs, the male and female take turns incubating. The male might incubate for, say, 6 hours. How does he know when his 6 hours are up? He seems to have an internal interval timer that measures that duration. Once it runs out, he initiates behaviors to get the female back on the nest. It measures a defined interval, not a cycle.
Fascinating. So a whole suite of timing mechanisms, from daily cycles to yearly ones to specific intervals.
Exactly. It allows animals to synchronize their internal physiology and their behavior with the many rhythms and demands of their environment in incredibly precise ways.
Well, today has certainly been a deep dive. We've journeyed from the intricate electrical circuits of the nervous system, how animals sense, process, react, and coordinate behavior, all the way to the subtle yet powerful internal machinery of biological clocks that allows them to anticipate and prepare for their world. It's just staggering complexity.
It truly is. These systems, shaped by millions of years of evolution, are masterpieces of biological engineering. The integration is phenomenal.
And considering all this, the localization in the brain, the plasticity, the autonomic control, these pervasive biological clocks, it really makes you wonder, doesn't it?
How so? Well, leads to a final thought, perhaps. If we connect all this back to ourselves, how much of our own daily experience, our moods, our ability to focus, our energy levels, maybe even when we feel hungry or sleepy, how much of that is being subtly orchestrated moment by moment by these internal timekeepers and these complex neural networks we've been discussing, often completely beneath our conscious awareness? That's a great question to ponder. What hidden rhythms, what neural whispers are shaping your day right now without you even realizing it?
Something to definitely think about. We really hope this deep dive has given you a new appreciation for the truly sophisticated inner workings of animal physiology.
It's been great exploring it.
Thank you, as always, for being part of the Deep Dive family.